All right. I'm going to introduce myself. I already introduced myself a little bit. My name is Allie Welsh. I'm a scientist at Foundation Medicine, so we're in Cambridge, Massachusetts. I don't know if any of you or many of you know much about what we do, but we're focused on genomic profiling for all types of cancer. And we started out by building comprehensive genomic profiling tests for use on tissue. Today, what I'm going to talk to you about is trying to apply that methodology to circulating tumor DNA. And so the goal of this session is really to think about new biomarkers, if they're useful, how to make them useful in the clinic, and what steps need to be taken to get there. So I hope this won't be too technical, but I wanted to share sort of how we think about the challenges with circulating tumor DNA as it compares to what we know really well with how to do genomic profiling on tissue, and how we sort of build a ctDNA assay with the patient in mind, with the clinician who may really want to use that assay. What do we need to do? How do we need to validate it? So just sort of to introduce, obviously genomics has really come a long way in the last number of years, and it has really helped us identify novel cancer genes at a really remarkable rate. So this is just the example of non-small cell lung cancer. What we understood in 2004 about the genomic makeup of this disease to now in 2017, a little outdated already, what we understand about the different molecular alterations that play a role in this disease. And you can see a number of different genes. You probably can't even read that from where you're sitting, but even within EGFR, there's lots of different ways that the EGFR gene can be altered, which may all have the same functional effect in making it active. And so there is this approach that we have used at Foundation Medicine, which we termed comprehensive genomic profiling, and others are using this approach as well now. And the idea is that this affords a lot of advantages for diagnostic testing and for understanding molecular alterations in cancer patients. And so the approach is designed to be tumor type agnostic, so you can use the same test regardless of what type of tumor, but it's really driven by an understanding of cancer genomics. So it's designed to be able to detect all 4 classes of alterations, so the 4 different ways that genes can be altered: single point mutations or base substitutions, short insertions and deletions, larger structural rearrangements and gene fusions, and also focal copy number changes and homozygous deletions. So it's a single test that can detect all of these types of genomic alterations at once, and it can detect them in many genes at once. And this is why it's designed to be applicable across tumor types. And so it's designed to sequence the full coding region, so all exons of all of the genes that it looks at. So for example, our original test is called Foundation 1. It's designed for all solid tumor types, and it looks at 324 genes. So you sequence the full exons of all of those 324 genes. And then it's also designed to be able to detect some of the more complex genomic biomarkers which are being used with increasing frequency. So we've talked a little bit about tumor mutational burden. Also, microsatellite instability can be measured using this test without doing additional testing. It's just different sort of algorithms that can be run on the data. A benefit of this is also that you don't have to know what you're looking for. Since you sequence all of these genes in the full coding region, you can find known common alterations, but you can also detect novel alterations that you weren't expecting to be looking for. And lastly, the approach that we use, which is a hybrid capture approach, is designed to have better sensitivity for these known and novel alterations than a PCR-based approach. And so I'm going to talk about applying this type of technology to circulating tumor DNA. But first, just to sort of set the stage in neuroblastoma, we have shown a little bit how this comprehensive genomic profiling approach can be used to understand and reveal what we know about the genomic landscape in neuroblastoma. So compared to what's been published extensively using integrative genome, exome, and transcriptome sequencing on a handful of 240 patients in neuroblastoma, the table below shows you, and the bar graph on the right shows you the results from using this comprehensive profiling test on 230 neuroblastoma patients, again performed on tissue. And you can see the common alterations: ALK, PTPN1, ATRX, MYC-N amplification, and MYC-N point mutations, and NRAS point mutations are seen at a similar prevalence as has been previously published and shown. But importantly, you can detect those different types of alterations. Even though MYC-N amplification is by far the most common, there's a few instances of point mutations in MYC-N. And similarly with ALK, even though fusions are the most common, even though base substitutions are the most common, there's also amplification as well. And so this comprehensive genomic profiling approach on tissue has, as I mentioned, facilitated the detection of known alterations, but also the discovery of novel alterations. And so on the left are just some examples of novel kinase fusions and novel transcription factor fusions that were detected in pediatric patients using this approach. So there's just one example there of an ALK fusion in neuroblastoma, but other alterations that were not necessarily expected that can be detected using this approach. This is just an example of 2 pediatric cases that showed a response to targeting these BRAF alterations that were detected. And then similarly, some other types of BRAF alterations that were detected. So these are structural rearrangements that were not known fusions, but that were still predicted to be activating. And this is, again, in a couple of patients, 2 neuroblastoma patients, where we found these rearrangements that seem to result in activation of BRAF as well, but these were not things that would be expected or necessarily picked up by a test that were looking for the most common types of BRAF fusions. So just sort of to give a little bit of background into why this type of profiling can have value in the clinical setting. And now we'll talk about how to sort of apply this to circulating tumor DNA. So there's a couple key ways that ctDNA really differs from tissue when you're thinking about biomarker testing. And the biggest of this is that a certain percentage of patients don't have any circulating tumor DNA at the time you take a blood draw. And many folks have published on this different amounts, but generally it's around 15 to 30% of patients. I would say on average about 20 across disease types and really across settings, 20% of patients just don't have any detectable tumor DNA in the blood sample. There's cell-free DNA, so there's normal fragments of DNA from healthy cells, but there's no tumor cells. So it's sort of the equivalent of getting a tissue slide that has 0% tumor cells and running your tests on it, but not knowing that it never had any tumor cells there. So we actually expect that 20% of patients where you take a blood draw and do a ctDNA test probably just don't have any alterations because there's not any tumor there. And of those patients who do have circulating tumor DNA, the percentage of that is actually really low. And so with tissue, for the genomic profiling test that we run, we require 20% of the cells on that tissue slide or tissue sample to be tumor. With blood, usually less than 2, often less than 1% of that DNA is tumor DNA. So the majority of it is normal, healthy DNA, and this means that tests looking for specific alterations have to be much more sensitive in blood than they are in tissue. And because of that, certain alteration types can be more challenging when that tumor fraction is super low. Specifically focal amplifications and some homozygous deletions if you're using this type of focused comprehensive profiling approach. And lastly, sort of at the moment, most experts and key opinion leaders will agree that tissue and ctDNA are sort of thought of as complementary at the moment. Neither can necessarily do everything, but certainly we're not in a place where ctDNA testing can replace tissue testing in the clinic. So when we think about building one of these comprehensive genomic profiling tests for use on ctDNA, there's a couple of key considerations that we need to think about. So as I mentioned, the amount of total cell-free DNA that you can get from a blood sample can really vary widely. So that's healthy and tumor DNA. Some patients have tons of this free-floating DNA in the blood, and some have very little. And we don't really understand yet who has which or what to expect at any given point in time. And so this means that we need to design assays to work from sort of worst-case scenario. Only if we get a little bit of DNA, we have to design them so that we can convert as much of that DNA as possible into sequenceable DNA. As I mentioned, 15 to 20% of patients have no tumor DNA at all, so we really have to think about how we interpret the results differently. And then second, that fraction of DNA that's tumor DNA is often really low, often much less than 2%. So that's 10 times less than what we normally see in tissue, which means we have to sort of be 10 times more sensitive with our assays. And so partially because of that, we really want to be careful not to further dilute that cell-free DNA in a blood sample with genomic DNA from the white blood cells or other whole cells in the blood. And so that's why collecting blood, if it's collected in EDTA tubes, it's important to process it quickly and extract the DNA. And if you're going to hold on to the tube of blood, it's important to make sure that it's collected in a tube that has a preservative. That'll keep all those intact, those white blood cells intact, and stop them from sort of opening up and spilling out the genomic DNA into the blood and contaminating that sort of precious fraction of cell-free DNA. So because of this low purity, we need to be able to detect mutations at really low allele frequencies, so down to 0.1 or 0.5% allele frequencies potentially. And because of this, when we do genomic profiling, we need to sequence much deeper. So the tissue test that we use, Foundation 1, that's used in a clinical setting, we usually sequence to about 500x coverage. But with the ctDNA test, we need to sequence to about 5,000x coverage, so about 10 times more. And we also need to bring the error rates of sequencing down using creative molecular barcodes and tagging methods so that we can make sure we can detect alterations at 0.1% allele frequency, and those are real, and make sure that they're not just noise introduced by the sequencing process. This means we have to redevelop a lot of the algorithms that we've used to make sure that they're optimized for these specific methodologies. Okay, this is a complicated picture and not really that important, but just basically showing you the workflow for doing this type of genomic profiling on ctDNA was really built off of what we already do with tissue profiling. So you get a blood sample, you extract that cell-free DNA, and importantly, you QC it to make sure that there is a fraction of this cell-free DNA. So this cell-free DNA exists at short fragments, about 150, 170 base pairs. So you want to make sure that there's a nice peak of that cell-free DNA and it's not contaminated by that genomic DNA. We attach fragment barcodes and we make a library out of it, and then we capture the genes of interest. So in the case of our ctDNA assay, it's 65 genes that we look for. So we capture the DNA from those 65 genes and we get rid of the rest of the library. And then that gets sequenced using 176 base pair paired-end sequencers on an Illumina sequencer to, as I mentioned, 3,000 to 5,000x coverage, so pretty deep. And then the computational algorithms will call all of those 4 types of alterations, so base substitutions, indels, gene fusions and rearrangements, and copy number changes on that data. And a report will be put together to provide to the patient that will list each of the alterations that was found and then the associated therapies and clinical trials. So without trying to get too technical, I just want to make one point because this is really a critical concept with ctDNA that tends to confuse sort of the field in general. So when we talk about sensitivity with ctDNA and how low of a mutation or how rare of a mutation can you detect in patients' blood, this really all depends on how much DNA we can get from that blood sample. So the process, we get DNA from a specimen. That little box there is just supposed to represent a gene of interest on a strand of DNA. That DNA is actually fragmented. And then we do this hybrid capture. So we bait for the genes of interest and we wash away the rest of the DNA. And then we do PCR on those fragments so that we can just expand the total mass of molecules that we have in order to sequence it. But that means we've created copies of each of those original fragments. And in reality, those are all mixed up in a tube, of course, and a subset of those are put on the DNA, the Illumina sequencers. And then after the sequencing, those individual reads are lined up against the reference genome, and we look at that gene of interest and we look for alterations. Now, in this case, you can see each of those fragments has many copies that are sort of PCR duplicates of the same original fragment. And so if we look at that one position in the genome where the dotted red line is, in this case, the total coverage in the example is 22x. There's 22 fragments of DNA at that location. However, a lot of that is what we call redundant coverage. There are these duplicate PCR fragments. And so this redundant coverage is actually important. This is what we use to correct errors. So we actually only call alterations that are present in every single redundant fragment, because errors that get introduced during the process that are artifacts often occur in only one out of these set of redundant fragments. So that redundant coverage is actually useful and very important. But on the other hand, we also refer to unique coverage. You'll hear that as well. And that means we get rid of all of the duplicates and we keep only one for each set of duplicates, so the one original fragment. And so in this example, there's actually only 6x unique coverage. That means there's really only 6 original molecules representing that patient's DNA at that site. So if we're looking for an alteration in a patient, an ALK mutation, for instance, we want to get a lot of those unique original fragments so we can find that rare, super rare ALK mutation. And if we only get 6 fragments, it's pretty unlikely that we're going to find a mutation that only occurs in 1% or 1 in 100. So this unique coverage is really what determines the assay sensitivity, and the more DNA that we can get, the higher the unique coverage we're able to obtain. So again, these two different types of coverage are both important. The unique coverage is important for assay sensitivity, and the redundant coverage is important for correcting errors, so it's sort of important for assay specificity. And so the ideal is actually a balance of both. And so this involves sort of tweaking the amount that you sequence the patient's sample based on how much DNA you get. And so the total coverage and the amount of DNA are the sort of the two levers you have to use to control that. Okay, so we've now built this ctDNA assay for use, designed to be used on patients. And we went about undergoing sort of a detailed analytic validation to make sure we understood how accurate is this test. Can it detect all these alteration types under real-world conditions before offering to use it on patient samples? And so I'll just go through a summary of how we did that in the next couple slides, but this was designed to sort of simulate all the types of alterations that we see or expect to see in patient samples and try to simulate them under real-world conditions. And then at the end, we look at actual patient samples and compare this test to other methods that are utilized, like beaming and PCR, digital droplet PCR. And we look at the results of these blood-based tests compared to tissue to see how accurate they are. And so the first step was really simulating a range of these alteration types using cell line mixes and engineered DNA, so synthetic DNA fusions that were spiked into healthy blood. So we mixed lots of different cell line DNA together at different amounts to try to simulate base substitutions, indels, and rearrangements, hundreds of each, at the range of allele frequencies we expect to see in patients. So this is just showing you that the x-axis is a log scale, so it's showing that we tried to bias the alterations we simulated to be a lot of these low allele frequency alterations that we expect to see in patients. And then the bottom plots there are showing you the expected allele frequency on the x-axis and the observed allele frequency on the y-axis. So we engineered all these samples, and basically we were able to detect all of these alterations with very high accuracy at allele frequencies that we were expecting to see. And this is showing you some data on detecting copy number amplification. So if you aren't familiar with looking at a copy number plot, on the x-axis are the different chromosomes, and then each of those dots on the plot represents one of the areas that we target with the assay. And so on the left-hand panels is a cell line that has 9 copies of HER2. So it's amplified for HER2 at 9 copies. And on the top, it's showing you 50% purity. In the middle, 20% purity, and at below, 10% purity. So that little star there is the HER2 amplification, and you can see at 50% purity, it's pretty easy to distinguish even by eye. When you get down to 10% purity, it becomes trickier. And so this cell line, you can detect it accurately down to 10%. On the right is a cell line that has 17 copies of HER2, so it's amplified to a higher degree. And in this case, you can detect the amplification down to 1%. And so just showing you that the ability to detect copy number amplifications is really dependent on how high of an amplification it is and also what purity of tumor DNA you get. So if it's a patient where you only have 1% of that DNA is tumor DNA, you might only be able to detect the higher level amplifications if you're using this type of genomic profiling approach. So this is just a summary of some of those experiments, not necessarily important to remember any details, but showing the sensitivity and specificity for each of these alteration types at the different allele frequencies. And again, the important thing is to do this on hundreds of samples so that we can provide confidence to the clinicians who are going to use this test on patients that, yes, this performs well, this is going to be accurate. If we find and we suggest that there's an ALK mutation in this patient, you can trust that it's real and it's not a false positive. And then the last thing that we did was compare two orthogonal methods. So we took the ctDNA assay and we ran it on 107 different clinical cell-free DNA samples, so blood samples from patients from a range of diseases. In this case, it was mostly breast and lung cancer, but we had a few others. And we compared the results to Foundation 1, which is our tissue test, also compared to digital droplet PCR and compared to beaming. And that's showing you the allele frequencies again with each of those methods on the x-axis compared to the ctDNA test on the y-axis. And it showed that all 128 alterations that were simulated were detected by the ctDNA assay. So that was good. That was what we would expect, hopefully, in comparing this test to methods that we know are well validated. And so lastly then, we've applied this to action. This is actually being used now in the clinic, and we routinely receive patient samples every day. And so this is showing you the data from the first 884 patients that were tested. On the left is a distribution of the tumor types. So we frequently get lots of lung cancer and breast cancer patients, but we have received a few neuroblastoma patients, and especially as part of our trials, which I'll talk about in just a minute. Maybe most importantly, we showed most of these past QC metrics. We're able to get high coverage, and we were able to provide results back to physicians within, on average, 12 days. On the right is just showing you the distribution of allele frequencies. So the inset there goes up to 2%. So you can see that the majority of these patients were finding alterations at really low allele frequencies, less than 2%. So as I mentioned, the tumor purity is very low. And this is just showing you the frequency of alterations in tissue compared to in blood. And so this is just the prevalence. This isn't in the same patient. This is a patient set where we had ctDNA and a different patient set where we had tissue profiling results, just looking at the general prevalence in lung cancer, breast cancer, colorectal, and prostate. And these are the most common tumor types that we see. So we just want to understand, is the prevalence of the p53 mutations that we're seeing in blood similar to what we see in tissue in these tumor types? And the answer was yes in these different cases. And then last, we looked at per patient comparison between tissue and blood. And so this was, I guess, 36 patients across a range of tumor types where we had a matched blood and tissue sample within 30 days of each other. So you would expect the genomics to be roughly the same. And in this case, 75% of the alterations that were detected in tissue were also found in the blood using this ctDNA test. 83% of those were short variants, so point mutations and indels. The challenge was copy number amplifications. Only 38% of what was seen in tissue was picked up in the blood. And 75% of the ctDNA alterations were found also in tissue. And so last, and maybe most excitingly, is a study that we've designed in collaboration with CHOP in neuroblastoma to use this test. And so they've accrued 24 high-risk neuroblastoma patients. 3 were newly diagnosed and 21 are relapsed patients, where they've been collecting peripheral blood for testing using this assay. All the patients are participating in various trials. Many are part of the Nepenthe trial, which is an umbrella trial using the tissue profiling or the tumor profiling to match patients to targeted therapy arms. And so many of these have temporally matched tissue samples that we can compare to. And they're also testing the blood longitudinally. So some patients have up to 6 blood draws taken over time. And the goal is really to understand how the results with ctDNA compare to tissue. Can we detect the clinical actionable alterations, and then how do these change over time? And so this data is going to be presented by Dr. Chris Bossi on Friday at 11:30. So if you are around, please check that out. It's really exciting data. Just a high-level summary. We found 94% of samples passed and yielded results. 76% of them had evidence of DNA or tumor DNA. And then we had 18 patients where there was temporally matched tumor and blood samples. And really interestingly, we found 54% of alterations were found in blood and tissue, but 43% were found just in the blood and not in tissue. And there was only 1 alteration found in tissue, tumor only, which was p53 alteration. So Dr. Bossi will go into much more detail on many of those cases during his talk, but this is a study that we're really excited about, and I think is specifically a way to highlight how we can start to incorporate these ctDNA tests into clinical trials and start to understand clinical utility. So just to summarize, there's really significant technical and biological differences with ctDNA than there is with genomic profiling on tissue. This requires really robust analytic validation of assays. And again, tissue is really the gold standard, but ctDNA can be useful in specific scenarios. And I think, again, as I mentioned, this collaboration that we've designed with the Children's Hospital in Philly is a great example of how you can incorporate these assays into a research study. There's some challenges for adopting these in the clinic, especially the fact that not all tumors shed this DNA. And right now we think of tissue and blood as really providing complementary information. And then last, cool things that are on the horizon, we're working on more immunotherapy biomarkers to be detected in blood. And ultimately, these types of assays will need to be improved and optimized if we're going to use them for early detection or for monitoring purposes. I think that's it. I'd like to thank many of the folks who are here from the CHOP group that we've worked with very closely on designing this study, as well as many folks at Foundation Medicine who have helped do all of this work. So thank you, guys.
Allie, do you have any data yet if you collect the sample after chemotherapy or radiation therapy, you're more likely to get cell-free DNA?
It's a really good question, and there was a lot of excitement at first. We thought that if you give a patient chemo and you're killing cells, you're going to be releasing lots of this cell-free DNA into the blood, and if you collect the blood, you'll get better results. That isn't necessarily the case. So the way that we measure alterations is an allele frequency, so the percentage of DNA that has that alteration. If you're able to measure it as mutant copies per mil, so the number of total copies, you might be able to detect it. So the challenge is when you give chemo, it generally kills a bunch of normal cells as well as tumor cells. So the amount of tumor DNA increases, but so does the amount of normal DNA that's kind of drowning that out. So if you have an assay that measures just sort of the relative percentage or the proportion of tumor to normal, it might not work. But if you had an assay that measured as a total number of copies, it might. So we've been working on ways to tweak that. But to date, no one's really been able to show how to improve the results right after chemo. And part of the challenge is ctDNA has a half-life of like 30 minutes in the blood. So timing it perfectly with when you give chemo and when you do the blood draw hasn't proven useful yet, but there's potentially hope there.